Hello, how are you? Here's part two, okay, of our cell practice, unit two cell stuff. Um, just another little story about lunch I had today. Guy near us was started talking as a nuclear physicist, and he was having fish and chips for lunch. See, that's like fish and chips, nuclear physicist. Oh, this is so good you can hardly stand it. So here's this question. This was a question one. This is the kind of question like the big question would be. It's got several parts. Here's some data. Here's a little diagram. So again, you're going to have to probably maybe go back and forth between these first two slides, but take a look at the questions. They're on the next page and come up with an answer and, pause it. And then once you got your answer, go to the next slide and I'll start running you through it. So here's all the questions. Pause and answer. Okay, so here's what they ask you to construct a graph. They're not going to ask you to do this, but if they gave you a graph, they might ask you a question because they told you they might about, like we had one before, identify the like dependent, independent variable. In this case, the variables, no matter what axis they are on, are the percent of ones that have this lap allele. That's what depends on where you got the sample from. So I don't think they could ask anything else about a graph, and they're not going to ask you to do a graph. So on to part B. Okay, describe, predict, right? Two things, 2 points, right? So effect of salinity on the frequency of the lap allele. That's the dependent variable, right? What's it depend on? Salinity, right? So here you have salinity going from low to high, and the frequency numbers go from lower to higher. So a direct relationship, salinity increases, that increases, or you could say the converse to get your one description point. Do not, again, give reasons in this part because you're just wasting your time. They didn't ask you to explain. They might later, but they didn't ask you to explain. Predict the frequency at a sampling site between site 1 and 2. Well, I already scribbled through it. Site 1 and 2 goes from 13 to 16, so your simple, you don't want to miss these kind, is between 16 and 13, right? Or you could say 14 or 15 or anything in between 13 and 16. Part C. Okay, so describe the most likely effect of this activity on the osmolarity of the cytosol. Describe the function of this in maintaining water balance. Now they're talking allobitum, osmosis, diffusion, and regulation. So again, two command verbs, both describe in this case, describe this, and then describe that. Okay, so what does it tell you up here? That this enzyme does. They remove amino acids from one end of proteins and release them into the cytosol, therefore increasing its concentration. The cytosol now has an increased concentration. Okay, so this is what this says, right? You could also say it in terms of water potential. And what increased concentration means is decreased water potential, right? Because remember, here I have these clipped off amino acids, right? And this enzyme causes there to be more and more of them. If this cytoplasm has more solute things, that means it has less water. What's water potential? It's the potential for water to go somewhere else. And if you've caused there to be less water in here, then there's less likely that that water will go out, right? Here's another thing about this. Osmolarity is a tricky word. Osmo lends your mind to think of water. Osmolarity is not water concentration, but solute concentration. Just like the adjectives hypo and hypertonic, they always refer to solute concentration, from which you can deduce the water concentration, but they always are solute. So the osmolarity, right, is going to increase as the solute concentration increases, but that makes the water potential decrease. Okay? Don't trip over yourself here and try to address all of those vocab words and say if it says to describe the effect, just pick one. Describe the concentration, describe the water potential difference, because if you contradict yourself, then no point, sorry. Okay, so how does this help these maintain water balance? Well, if it's real salty out here, right, and it showed before in salty water, the concentration of the, or excuse me, the frequency of this gene and therefore this protein and therefore the concentration of it increases, that means it's less likely that these things are going to lose water because their cytoplasm has become more concentrated. So prevents water loss to the environment is all they're looking for. Again, no explanation necessary. Don't go into why, right? Save time, don't go into why. If you're done with this part of the test and you still have time left, it wouldn't hurt you to go back and add why if you want to, but pay attention to these command verbs and do what they say. Ready for the next one? Part D, okay. It says that the larvae of these things, that's a key word, are evenly dispersed, even though that whole diagram was the fact that the grown-ups are not evenly dispersed. You find more of them here and less of them there, right? So as they mature, they attach to rocks, and then they don't get dispersed around by the current, the water movement. So explain the differences in the frequency among the adult populations at the sample sites, despite the fact that the larvae are all around, right? Why do you find only the ones with high frequency in salty water and with low frequency of that gene in less salty water? That's what they're asking, right? So explain the difference. If they have this allele, they're more likely to survive in that high salinity because they are not going to lose as much water. That's what we just said in part C, right? Or you can say the converse side, the ones without the lap allele are less likely to survive in the high salinity. That's why you find less of them there, right? So now they ask, okay, let's change some things up, right? Let's say that the lap allele was found in all of these mussels, not just in some. What would be the effect, do you predict, on the distribution of where you find these in Long Island Sound? Not in the ocean, but just in the Sound. The Sound, if you remember, had a part that was closer to the ocean, saltier therefore, and less close, less salty, right? So what would you think? Well, if they all have this ability to tolerate salty water, you'll find more of them in salty water. Or you'll find less of them in less salty water, right? Justify your prediction, right? 3 points, 3 command verbs. Justify my prediction. I already said it pretty much. The ones in the high salt with that lap allele, they will be able to osmoregulate, right? So it's not just enough to say that you'll find mussels with the lap allele and high salinity. You already said that over here. Now they want why, and that is because they will be able to osmoregulate, and the ones in low salinity will not osmoregulate. This is a good one. So again, take a gander at this and give it a shot. Pause to make your answer, and then shoot me back on. Okay, so if we look at this diagram, okay, we have a cell that they tell you is a plant cell, right? We have that big central vacuole, and we have some membrane proteins in both the vacuole, which is called the tonoplast, the vacuole membrane, and the cell membrane. This one's a proton pump. They tell you that. This one has protons going this way and potassium that way. And this one has potassiums going through it. It's a channel. It's passive transport. They tell you this one's active, right? So here we go. Identify the cellular component in this model that's responsible for the increase in pH. Oh, we got a little pH stuff here too, right? Increase in pH, they expect you to know that pH is about protons. More protons, lower pH, more acidic, right? More hydroniums, more acidic. So what has happened to make an increase in pH must mean it has become less protons in there, right? In where? In the vacuole, right? When flowers are opening, how did the protons get out of the vacuole to make the pH increase? They diffused out this way. Or, excuse me, they got pumped out this way, actively transport pumped. Okay? So that's the identification of that transport protein that's responsible for that. And then describe the component's role in changing the pH. That again, I know that pH and protons have to do with each other, and how it did it, its role was to move them out of the vacuole. A researcher claims that the activation of this transport protein causes the vacuole to swell with water. Now we're back into the osmosis kind of thing. Provide reasoning to support it. Support what? He already tells you that the vacuole is swelling with water, so you don't need to say that. You need to explain why, right? Which would be his reasoning to support it. Okay, so what happens when these protons get pumped out? The potassium comes in. What does that make this? That makes it hypertonic, right? Hyperosmotic, more osmolarity, to use that word. And what's that with water potential? Lower water potential. Lower potential for water to go out of this, which is why the water's coming in it, like this arrow shows, right? That was his claim. That's the reason he made the claim. Bingo. There you go. Another sunrise shot from this morning to leave you with. I hope you did well with these practice ones. Please give me some feedback. And good thing tonight, if you watch this today, if you happen to, tonight, this is Tuesday, on the Gene on PBS, which I sent you an update about. Should be really good. Have a peaceful, distant, healthy rest of the week. I'll touch base with you for the rest of the week. Bye-bye.